I would like to thank the organizers for giving me the opportunity to share our preliminary findings on the impact of microgravity on gene expression. Galaxies like ours is filled with areas of microgravity and super hypergravity like black holes, which they collide and produce supernova. Supernova is one of the most catastrophic events in our universe. This is not a galactic event, but a picture of a fibroblast-like cells with its stained actin filaments and nucleus. You can surely imagine the similarity of the two images, even though their scale is way different. Stem cells are inherently designed to maintain their numbers in any given organ and only divide when there is demand. Unlike differentiated cells, they undergo asymmetric cell division, thereby one half of the daughter cells remain identical to the parent stem cell, and the other half proliferate and differentiate to give rise to specialized cells, which are needed for repair and regeneration. Because it is so difficult to expand stem cells in the lab, we ask the question, can stem cell expand in microgravity without or limited differentiation? Basically, can microgravity induce symmetric cell division? As we examine this, we want to understand the impact of microgravity on gene expression. The overall objective is to assess the feasibility of generating clinical-grade stem cell in microgravity condition at the ISS environment for human use on Earth. In essence, we want to evaluate the safety of using space-grown cells in regenerative therapy with regards to genomic integrity and stability, tumorigenic potential, as well as potency and efficacy of the cells.
One, and lift off from the Falcon 9 to the space station.
I couldn't be more excited on the launch date because we have been in the queue for nearly 2 years. Cells were seeded and cultured in biocell cassettes. Astronaut Peggy Whitson worked on our project at ISS. I always say she was the most expensive lab tech I have ever had. We transported and culture expanded 3 types of cells at ISS. We grew non-adherent cord blood-derived CD34 positive hematopoietic stem cell. We also grew non-adherent leukemia cell line K562. This is a well-known cell line that has been used for many years. And finally, we had also grown bone marrow-derived mesenchymal stem cells. Identical ground control were also set up in our lab. We received near real-time imaging from ISS beamed down to Earth. Imaging started at 3 days before launching and up to 13 days after launching. The ground control cells were also imaged simultaneously in our lab. Real-time images of hematopoietic stem cell at ISS compared with ground control showed significant increase in numbers without any obvious morphological difference. The K562 leukemia cell line appeared to form clusters, as you can see here, at ISS compared to the ground control. MSCs at ISS show no obvious differences in morphology and proliferation rate when compared with ground controls, even though the cell projections at ISS appear to be larger, but this could be an imaging artifact. Cells were cryopreserved in 5% DMSO, in RNA later, and in formaldehyde. They were kept at minus 95 degrees centigrade on the ISS freezer until they were returned to Earth using a SpaceX return capsule that landed in the Pacific Ocean. And the cells were, the capsule was identified, and the cells that are on payload cargo were shipped to us by FedEx within 24 hours. You could imagine, after such journey through space, you get the cells sent to you by FedEx. But anyway, they came in good condition, so we were glad to receive them. Once we received the cells, the real action started. In our lab, the space expanded cells under ground control were carefully characterized. We've done a number of studies to establish their identity, purity, sterility, post-flight, and also to assess their proliferation rate. We have done also DNA damage studies and tumorigenesis, as well as assess their regenerative ability and repair. Some of our findings, especially the ones for the MSC, has been reported in this Nature microgravity paper. We are yet to complete our analysis on hematopoietic stem cell and K562 cell lines. So stay tuned. Cells preserved in RNA protect were subjected to RNA isolation and real-time PCR for cell cycle checkpoint gene expression analysis. For MSC, which we also reported in the Nature microgravity paper, we examined the impact of microgravity on CDKN2A expression. CDKN2A is a G1/S1 checkpoint inhibitor. We also evaluate E2F1 expression, which is a protein that is known to impact cell cycle progression, and it is known to bind, to preferentially bind to retinoblastoma protein in a cell cycle dependent manner, thereby promoting entry into a space. And finally, we look at follow-like kinase 1, which is a serine threonine protein kinase that facilitate transition from G2 to M2 phase of the cell cycle. PKL1 promote maturation of centrosome and establishment of bipolar spindle. MSC were cultured for 1 to 2 weeks duration. Compared to ground control cells, the expression of CDKN2A was not significantly different in space-grown MSC after 1 or 2 weeks in culture. This suggests that within 7 to 14 days, microgravity has minimal impact on MSC capacity to enter into the S phase of cell cycle. Our analysis shows that E2F1 expression by space-grown MSC may be decreased in microgravity environment, although this decrease was not significant at 1 week or 2 weeks culture. Our analysis showed PLK1 expression by MSC in culture was decreased in microgravity environment, but this decrease was only significant after 2 weeks in culture. In summary, it appears in a 7-day culture, microgravity does not significantly alter the expression of CDKN2A, E2F1, FLK1. However, within 14 days culture, microgravity appeared to downregulate the expression of PLK1. Therefore, microgravity conditions may slow the progression of MSC at later stages of cell cycles during long-term culture, but this does not appear to reduce their overall growth rate relative to the ground control. Further studies would be needed to fully validate this finding. We have performed RNA-seq differential gene expression analysis between sample culture and culture expanded on the ground or at ISS for all the cell types that we sent to space. The table showed the total number of the genes expressed in all the samples with no filtering. This was found to be 15,368. Depending on the cell type, the time period in culture, and the gene filtering criteria applied, the number of genes that are differentially expressed between ground and ISS significantly varies. As you can see, it appears that K562 leukemia cell line was the most impacted, followed by hematopoietic stem cell and then mesenchymal stem cells. The more stringent the filtering criteria, the less the number of significantly differentially expressed genes. This is fantastic result, but what does this all mean? How do we make use of this knowledge to further advance our understanding of impact of microgravity on human physiology? This is where pathway and network analysis comes in. The goal of pathway and network analysis is to characterize biological meaning of the relationship among genes. One can start with a list of genes or a gene expression table. And there are common tools for pathway and network analysis. This can be commercially available software such as Metacore, or pre-tools such as weighted gene co-expression network analysis, WGCNA, and others. The statistical basis of a given pathway tool, such as Metacore, is the use of hypergeometric tests, such as Fisher exact test, chi-square test, to test if members of a gene list are overrepresented in a given pathway or gene ontology categories. The key to pathway analysis using these tools is to find enrichment. Pathway significance depends on key factors: the total number of genes with measurable expression value. So as you know, not all genes from our experiment have measurable expression value. So therefore, gene filtering based on expression threshold is needed, and that's the first step. Also, the number of significantly different genes needs to be established using statistical tools such as t-test, ANOVA, and multivariate linear regression analysis, which is the second step. And finally, using the platforms like Metacore to do the pathway analysis. We have examined the microgravity impacted genes based on how they associate and enrich with gene ontology processes. The enrichment or association was ranked based on negative log p-value. In the figure, hematopoietic stem cell culture at 1 week is designated with orange color, while K562 at 1 week is given blue color, and MSC cultured for a week with red color, and finally MSC cultured for 2 weeks with a green color. Both hematopoietic stem cell and K562 rank the highest in pathways associated with positive regulation of proteins localization to membranes, while MSC seem to associate with pathways involved with regulation of trophoblast cells migration. I find the association with diseases to be the most interesting, particularly the association of microgravity impacted genes or MSCs in relation to muscular dystrophies and atrophy. It is interesting to note the longer the MSC were in culture, the stronger the association with muscular dystrophies and atrophy. We all know the impact of long-term space flight on astronauts, particularly with regards to muscle wasting and atrophy. On the other hand, our finding may shed more light on the mechanism of muscular dystrophies and atrophy and how they may manage, how we may be able to manage or treat such potentially fatal diseases on Earth. So far, we have focused on pathway analysis, but sometimes the genes of interest may have connection beyond canonical pathway boundaries. So network analysis is employed and is generally characterized by node, which are the genes, and pathways, which are the link nodes, and then networks, which are the interconnected nodes without pathway boundary. Network analysis can be identified, can identify connection beyond pathway boundaries. Metacore network analysis is based on pairwise interaction database. This pairwise interaction database is collected from literature and it kept growing as more interactions are found. Then different algorithms are used for the network analysis. Another tool, statistical tool for network analysis is weighted gene co-expression network analysis, known as WGCNA. This is aimed to build networks with scale-free topology, such as the use of hop genes, which are the master key regulators, and then it also characterized by robust non-hop genes that are altered. It is also sensitive to targeted attacks on the hops genes. So for example, the association of microgravity impacted genes with cells adhesion and cell matrix interactions in our data set, we discovered gene candidates like TIM1 and galactine that stood out. They stood out from the rest of the connections and pathways. And then clearly there are other potential hub genes as well. These hub genes that we identified in our data set were organized based on whether they are extracellular, membrane-bound, cytoplasmic, or nuclear in terms of their localization. we have begun studies to validate and fully characterize and understand the role of each hub gene under microgravity condition and how it may relate to the impact of microgravity on astronauts and diseases on Earth. In summary, using pathways and networks analysis, we have discovered key pathways impacted by microgravity. We also identified a number of hub genes or master regulators for further studies. We have identified genes that can be used to further understand the impact of long-term spaceflight on astronauts. And these genes has been also associated with muscular dystrophies and atrophy, which may shed more light on the mechanism of such diseases and generate possible new therapies. This is a team's effort, and I would like to acknowledge members of my lab, particularly Dr. Pang Huang, who has done most of the experiments that I presented in this, and our collaborators from outside of Mayo Clinic, Larry Harvey from CAST, and our implementation partner, which is BioSAP, affiliated with the University of Colorado. Thank you so much